we finish the replication, especially the recombination section, and then little bit of touch base on the DNA domain, and then we move to transcription. Can you please close the door? Thank you so much. So, replication we did. You need to know the basics of DNA replication, the chemical reactions that happen during DNA replication, proofreading, the enzymes involved, correct? All this stuff. So, and replication for. So once you did it, we briefly touch base eukaryotic transcription and also the telomeric DNA replication. Telomeres and telomerase function, how telomerase helps replicating ends of the chromosome, correct? And then we go briefly did how the RNA genomes are replicated basically using the reverse transcriptase function of AGT, the antiviral drug, and I think that's it. Then we will go into the recombination briefly. So you already know the basics that recombination is nothing other than coupling the DNA. Sometimes it is necessary for the defense mechanism, sometimes it is necessary for supplying the parental genome into the offspring, correct? That's why during a meiotic recombination there is changes in the DNA sequences, not the sequences, the recombination. So that's why we are different than parents. Otherwise, if there is no recombination, we will be identical, correct? So genetic recombination is nothing other than coupling the genetic traits, alleles, or the DNA. Genetic recombination rearranges genetic information. Sometimes recombination is used for repair if there is a bad genome or mutated genome. It can be deleted off by recombination, and there is something called transposition where sometimes the viral genome recombines or a foreign genome recombines with a host genome. These are all recombination events. So in those cases, integration of the foreign genome, and that's part of the molecular evolution. Sometimes recombination, rearrangement could be nasty. Okay, like some of the leukemia. In fact, my research lab works on something called mixed lineage leukemia, where one of the genes, which is a critical regulator of hematopoiesis. Hematopoiesis means blood cell differentiation. We have bone marrow which differentiates to give you the different types of blood cells, correct? And this particular gene plays a critical role in normal bone marrow differentiation. But for whatever the reason, if there is a rearrangement, yes, you have some question?
Oh, what's the last word of the
Leukemia, mixed lineage leukemia. You don't have to know all of it. I am just bringing it as an example. So this gene is pretty big, okay, but for some kind of pressure, it's not atmospheric pressure. For it could be chemical stress or some kind of stress. This gene breaks down somewhere, and this recombination event takes place where it recombines with other part of the other genome. So this particular gene is critical for blood cell differentiation. If there is breakage or rearrangement or recombination event, it cannot normally function to produce normal blood cells, and that's leukemia. Okay. So often the people who are professionally exposed to benzene, okay, those who work in coal mine, purification of these organic aromatics and so on, some of these people actually develop leukemia. And one of the reasons of this leukemia is that this high concentration of aromatic compound exposure leads to the breakdown of these. So basically these aromatic compounds, once they are exposed, they go and bind the DNA, and that puts some kind of chemical stress and forces the body to recombine, go for recombination event, and that causes leukemia. So I gave an example, but there are many such events. Recombination could be bad. So recombination means reshuffling of the genetic information. Most of the time it is good, correct? Mitotic recombination, deletion of abnormal DNA, but sometimes recombination induced by certain chemicals or stress could be problematic. So compared to the parents, the progeny may have new combination of traits. You have same DNA sequence, but they are reshuffled, correct? They are at different positions. So position in the genome has a significant impact how it functions. It's not only the DNA sequence. So there are multiple type of recombination. It's a very big area of research. Lot is known, and but still it is complex. So sometimes it is called homologous recombination. If two genes are similar, okay, if two parts of the DNA, two chromosomes or two DNA is similar, okay, they have similar, basically sequences similar. These two DNA can go to recombination. So this part, let's say, okay. So by recognizing this homologous region, it can actually, this part to go there and this part to go there. This is very well studied, something called homologous recombination. So two chromosomes or two genes to the DNA molecule which are similar to region, they sometimes cross over. Okay, and this is called homologous recombination. We will talk briefly about this, how it happens. So homologous recombination is kind of, one understand that they are homologous region. Recombination involving similar DNA sequences are called homologous recombination. And sometimes some recombination does not leave this homologous region. These are called non-homologous recombination. There are many proteins responsible for the recombination event. Basically, if you have to combine these two, you have to break this DNA here, break the DNA here, and then combine, correct? Ligase. So there are unique endonuclease and variety of enzymes. Some of the recombination proteins, something called REG-A. Rake means this protein is involved in recombination. That's why it's called rake. Recombination associated protein A. Possibly this protein was first identified, which helps genetic recombination. So people named it rake A. There is rake B, rake RAB A, RAB B, RAB C. These are also involved in recombination. So recombination, this is a very well established model for homologous recombination, and this is what I want you to know. It's conceptually very simple. Okay, and well studied, and lot of enzymes or mechanism has been discovered. Can you see this well, or I will expand it? This is called Poly-A model of recombination. Is it good? Good? Can you see? Better? Yes or no?
Yes.
Okay. So you have two molecules. It is color coded, so that's why it is easy to understand. You have two DNA molecules, and these two have some homologous region somewhere here. Okay. So what happens because of the homology? Once there is a nicking on each strand, one of the strands here nicked and here nicked. So because of the nicking and homologous region, this particular strand is complementary to this particular strand because they are homologous, correct? They are similar DNA sequences. So basically, the first step is that one of the strands is nicked, and once nicked, this particular strand is exchanged with the strand on the other DNA, which is similar sequence. So they are correct, similar and complementary. So nickage, first is the nicking, so there is the breakdown. Then there is strand invasion. Because of the complementary sequences, this DNA now can complement to this particular blue one. Does it make sense? Because they have the similar sequence. So similarly, this complementary strand is also complementary to the red one. So it's kind of exchange. Does it make sense? Once it is exchanged, it gets filled. So this is called strand invasion. So strands are exchanged. Once it is exchanged, so opposite strand comes here, it seals here, basically ligation. The other strand seals here, ligases, and it produces a junction. Does it make sense? So you need to find the complementary sequences, homologous sequences, and these homologous sequences come close. One of the strands is cleaved on both the duplexes, correct? Then, because of the complementarity, there is a strand invasion. So opposite strand comes here and ligate, and that produces a crossing junction. And this, once it is ligated, this junction can move either direction. Okay, so it favors by the complementarity. So this junction or the branch, let's say, it moves this direction. So here is the junction. Originally you were here, now you are here. Correct? But this particular DNA comes from the other chromosome. This part of the DNA comes from other chromosome, this part of the DNA comes from the other chromosome. So there is, correct? Reshuffling of the DNA from two different duplexes initiated by homologous recognition, homologous sequences recognition. So now under this situation, branch has migrated here. Now you can just take a scissor and cut it here. Correct? Cut and like it. Now if you cut here, now this particular, the original you had one blue and then whatever it is, light blue. Now this part of the genome, so you cut it here and seal it. So this part of the genome, DNA sequence, comes from different chromosomes. Does it make sense? It's conceptually. So take a scissor and cut it here. This is simple. Correct? So you have an exchange of DNA from another chromosome. This is a recombination. These are of course all catalyzed by enzymes. So there are branches recognized by certain endonucleases. So endonuclease cleaves here and then lies. So sometimes you could, here we are doing the west side and east side cleavage, but sometimes you can also cleave here, this side. So this is done a little bit extended here. So this junction actually does not look like a straight line, but it is more like a scissor, correct? Cross. Now you can cut here. This is the original strand. This is the invaded strand. So if you cut here, this part, west and east, if you cut it, you get, correct? You get this one. So your original strand is here, then this part. So if you do the west and east cleavage, if you cut here and here, your this strand stays the same, which is this one. Now you have this part, this section is this section, and this section remains the same. Correct? This part is new. Similarly, the other strand will be red. This section is new. Is it clear? So this is east-west cleavage. You could also cleave north and south. If you cleave it here, you have a different combination of the threads or the DNA sequence. So both east, west and north, south type of cleavage has been observed, and that gives you different combination of threads. This model is called the Holliday model, based on the name of the discoverer, that Holliday model of for the homologous recombination. So this is initiated initially by homologous recognition. Any foreign gene or any two genes, when they need to recombine, homologous recombination is one of the major choice because they can easily come close together, correct? And then need strand invasion, branch migration, east to west or north south cleavage, you get different recombination products. I am not going detail of the lot of enzymes involved, but as soon as you see that REC A, REC B, REC C, D, correct? These are recombination related proteins, and they are involved in this breakdown event. REC, RUB, these are recombination related proteins. If the concept of homologous recombination is clear, so I may ask in your exam, draw a cartoon explaining the homologous recombination, something like that. Can I do? No? Why? Why do you have problem? Huh? You still have problem understanding that? Then why cannot ask? So what should I ask? You tell me. If you understand, then I should ask it. So it's easier, correct?
Weirdly, what we're supposed to be able to draw.
The model of holiday model of correct homologous recombination. Know the name of the enzymes, proteins involved in recombination. Sometimes, actually, you design strategies to force the recombination. Okay? Any of you heard the name called CRISPR? CRISPR technology is I think it may get Nobel Prize if it turns out to be useful for the therapy. So there are many events, many diseases. Sometimes you see one of the gene is malfunctioning. Okay, one of the gene is malfunctioning because either the gene is not there or it's mutated. Correct? Let's explain a little bit. Something called Angelman's syndrome. Anybody heard? It's a neurological disorder. The kids, especially who have this type of problem in the childhood, they are abnormally happy. If there's some young kid is happy or all the time smiling for no reason, then there is some concern. Rare, but it happens. That's why it is called Angelman syndrome, because the kid is like an angel, correct? All the time smiling. But it's a neurological disorder, and there is a gene called UBPA. You don't have to know for your exam, so don't worry about it. Just know, correct? UBPA is a gene which is critical for neuronal development. Correct? Neuronal function, neuronal development. And when we get the gene at the childhood, we get one copy of chromosome from the father, one copy of the chromosome from the mother. So one is paternal copy, another is maternal copy. We get one copy of UBA3A from mother and one copy from father. Does it make sense? But for some reason, nature has designed that the paternal copy of the gene does not get expressed. It gets silent. Okay? Although we have the gene, it does not get expressed. So it's called, anybody knows the name of the phenomenon? Imprinting. It's called genomic imprinting. There are many such instances. If you take my other classes, advanced classes, I go in detail because these are epigenetics. Correct? You have the gene, but it's not working. So the environment of the gene is not right, so that it is not expressed. Now we are left with one copy of UBA3A from mother, but for some reason, if the maternal copy is not functioning properly, it's mutated or deleted, the kids are born without active UBA3A gene, which is critical for the neuronal development. Does it make sense? It's a ubiquitin ligase involved in protein degradation and so on, signaling pathway. So this gene is mutated or deleted. So what to do? There is no therapy. There was no therapy for a long time. But now, with the CRISPR, this technology, this is actually a very interesting technology. It uses the bacterial defense mechanism where you can actually design a targeted recombination event. So you can have a vector where you can load a correct TB3 gene, it's correct, and then this gene you can target to replace the mother malfunctional gene with correct TB3. Does it make sense? So basically you are supplying a foreign gene, and this technology helps you to design a strategy where this foreign gene can recombine specifically on this particular location. Does it make sense? So now we are able to correct the genetic mutation or replace the wrong gene with the right gene. And if we are able to do that, so if it is detected early enough in the childhood that there is a problem in this particular region of the gene, we could actually correct it at that stage. Right now we could do that in a cell, sometimes in animal, but doing in human is still not so easy, correct? Especially if we do something in the human, correct? We don't know what is the other impact. So that's why there is a big debate how we are going to do it. Unless you are extremely comfortable doing in the animal or the chimpanzee or the dog, correct? Monkey, pigs, people are not going to try in human. But there are certain diseases, genetic disease where there is no therapy, kid is going to die. In those circumstances, people do consider for clinical trial with this risky stuff, correct? And I think one or two instances people think that there is a high success where people never thought there is any possible therapy. Okay, that's the CRISPR technology, and if it is successful in human, right? So many of the non-treatable, correct, genetic disease could be treated with this guided recombination machine. This is basically guided recombination vehicle. Does it make sense? So recombination in this case will take the advantage to treat genetic disorders. So like this leukemia where the MLL is mutated, if we have the recombination technology to correct the MLL gene, we could do. In fact, one of my early PhD students, he went for postdoc in Harvard Medical School. Right now, he is working in a company where they are actually redesigning the stem cell, bone marrow stem cell, with this CRISPR technology to supply the correct gene. Correct? So these are a lot of interesting stuff going on. But for your class, don't worry. For the exam, don't worry about it. Okay? For exam, know that the recombination happens, polygon model of recombination, some of the name of the common recombination related proteins. Does it make sense? Okay, so holiday model of recombination events. Lot of interesting stuff happens, especially in the immunology area, because our immune system reacts to different pathogens, different antigens, correct? Abruptly and so specifically. There are a lot of these recombination plays a critical role. So we are not getting into the immunology in this class as well, but recombination is very important. I skip all this stuff. So we discussed basic principle of homologous recombination, and then there are other modes of recombination like non-homologous recombination and so on. Now, one of the application of recombination could be it is used for the defense mechanism or repair. Now, when there is a need of repair, that means there is a normal DNA sequence is mutated or damaged, correct? We took Florida sunbath, correct? Very often, and that helps to develop skin cancer, right? So that means if we are spending regularly, there are a lot of UV radiation, correct, in the sunlight, and this UV radiation, if it is, let me see if I have that. Yeah, this is one of the mechanism by which UV can damage DNA. You have the two adjacent base pairs, correct? They can cross-link with each other. They can cross-link by radical mechanism. UV light triggers the radical reaction, correct? You have studied in organic 1 or organic 2, correct? How to activate CH bond for chlorination. Let's say you take methane, you want to go to chloromethane or chloroform. What is the chemistry?
Halogenation? Is it?
Yes?
I don't know.
CH4 to CH, 3Cl, CH2, Cl2, CH, Cl3, and CCl4. What chemistry do you use?
Free radical halogenation?
Free radical halogenation, right? You can sunlight, you can UV light and put the chlorine, it produces radical, right? Whenever there is this high energy light, it's bad. That's why when you go for X-ray, right? They cover the rest of the body so that X-ray cannot fall in. Because again and again, X-ray could damage and could lead to DNA damage, free radicals. Free radicals not only damage DNA, it also damage other lipids, membranes, proteins, and which can have dangerous impact. But if it impacts the DNA, that could be a major problem. Now, this is a cross-linking between two adjacent bases. So this DNA cannot be replicated because two bases are crossing, correct? So this has to be repaired. So UV radiation is a problem. Many times UV radiation, high intense energy, is also used to kill the tumor cells. Correct? Somebody has tumor and irradiate with high energy light, that will damage the cells, including DNA, the tumor cells will die. So, anyway, we are always exposed to certain amount of UV light, so there is a potential of this DNA damage. This is one, but there are many others. So this damaged DNA, once there is DNA damage, the cellular machinery is designed to recognize the damage. First job is to repair the DNA, remove the damaged part of the DNA, and replace with the right DNA, so that there is no mutation. So, if cell cannot mutate, cell cannot repair the damaged DNA, the next job of the cell is to die. Otherwise, correct, we will be living with damaged DNA, and it can cause cancer. So, which body physiological homeostasis mechanism do not want. So, they eliminate the damaged cells. Correct? but some of the damage is tolerated by the system. That's a problem. When there is a damage, it cannot be repaired at the same time, right? Cell does not commit suicide. So these cells are alive with damage and they carry on. So basically, we live with mutation. And if there is mutation in certain critical part of the genome, or depending on the how much total mutation we have, that may imbalance the cell proliferation and contribute to tumor. So those are the balances, whether cell will be differentiated or will be tumorigenic. That depends on the type of mutation and amount of mutation. Correct? So all these things. So that's why cancer is one of the worst disease. We cannot cure it because we don't know what is the cause of the cancer. Does it make sense? If it is a bacterial infection, you can treat with an antibiotic. Bacteria will die, we survive. But if it is a cancer, we really don't know what caused this cancer. What we see is what happened after this cause. Does it make sense? So we always try to slow down the cancer or remove the tumor or do something to the cancer cell. We rarely have the opportunity to treat the origin. It cannot be cured most of the time, the 100% success, until we cut out the organ and things like that. Does it make sense? Even then it is problem. So that's why I think this is one of the most difficult disease to develop therapy. So this is one example of mutation. There could be chemical damage. I gave you an example of chemical damage. The occupational exposure to aromatic molecules like benzene. When you work in organic lab, we suggest you or we advise you, correct, that you do all the organic reactions inside the lab, inside the hood. That's the hood the design. But many times I sometimes like to visit the organic lab. I see the student is putting his head inside. Correct? You have a barrier, there is a reason. Correct?
Right.
And instead of that, student is putting his head inside the hood and trying to do something. That's actually dangerous. We ask you to not to throw out these organic solvents outside, especially the aromatics, because they can generate free radicals very easily and that can cause problems. Right? Anytime you ask the question where these gases go, which are taken by the hood, let's go out. Correct? Eventually, some way there we are getting them back. Correct? Those organic, inorganic waste. If you think about where they are going, then we are not doing the right job. Eventually, they are dumped somewhere. Correct? They are contaminating the water, contaminating the ocean, and things like that. Correct? The drinking water, what we are drinking, has actually many of these components which we are disposing, including the pharmaceutical. Correct? We buy a lot of medicine, and once the use part of the medicine, and I think about 50 to 60% of the medicine goes to the brain. Am I correct? So those medicine eventually contaminate the groundwater. In fact, if we detect our water, there are trace amount of each of these medicines, and many of them, right? And we are exposed to some of this stuff. So eventually these chemicals, if they go reach to DNA, or sometimes they don't need to reach the DNA, they can induce DNA damage and mutation. Correct? So chemical damage. How many of you run DNA gel in a biochemistry lab? What How do you visualize the DNA? You know how bad Ethereum bromide is, right? It immediately goes to the DNA and it can mutate. That's why we say, don't touch the solution, it's bad. Don't dip the droplets of the buffer on the floor, but still, students think that research is more important, forget about life. Correct? Wow, DNA, nice gel. Take a picture and carry in hand and things like that. When I was a PhD student many years ago, I was purifying a compound. So organic, I did a PhD in organic chemistry, so we made some molecule. This molecule was only separable by organic solvent like benzene. Correct? And my PhD supervisor did not do the right thing to me because I was analyzing couple of months constant amount of benzene, open lab. And I blame him right now, but he's a big man. He cannot do much. So, but I don't tell the same thing to my student because I understand what is the significance of benzene, how it would. Organic chemists sometimes are crazy. Don't tell the organic chemist here. I am an organic chemist by training. So they don't, they think, wow, these are molecules. Very nice. Correct? Nice crystal smell. I do sometimes. Correct? When we are making the molecule in a test tube, I made the molecule. I smell. Don't do that, okay? I try to do that. Yes. But these molecules sometimes could be nasty and one has to be careful. The metal ions, the lead poisoning couple of months ago, maybe a year ago in Michigan, in Flint, correct? The water was contaminated with some metal ions. Was it lead? Lead contamination? So lead is one of the lead 2 plus, right? Why it is bad? Because it's a plus 2 charge and it goes and binds to DNA very easily. Alright? It can also bind to many metals. Lead is fantastic for coordinating the thiolated molecules, enzymes. They block the enzymatic function, they block the DNA and damage DNA. Arsenic, correct? Arsenic 3 and arsenic 5, there are 2 forms, and one of these forms is pretty dangerous, especially people. Arsenic, there are some county in this country, some area there is problem, but it's a major problem in this Bangladesh, some part of India, Pakistan, especially the Himalayan belt. Okay. Under the underground there is a layer of arsenic, but people in remote areas, they don't have water supply. So they drill all the way down to get the groundwater. Instead of getting a good groundwater, they think water is contaminated on the top, so get the groundwater, and this groundwater is contaminated with arsenic. Instead of safe drinking water, they get infected poison. And eventually, these people have tremendous health problem, and politicians are happy, okay, giving them some money to drill a seawall, but don't worry about that. So these are some of the cases where your metal ion could be problem, and they could damage DNA because they can bind to DNA positively charged and they destroy the protein function and all kinds of stuff. I think arsenic 5 is used as a poison to kill the tumor cells. AS2, I think AS2 or 3. I think this one or the other one, arsenic oxide, is used to treat leukemia. It's a chemotherapeutic drug because it kills the tumor cells. So many of these mutagens could be organic, aromatic, metal ions, and whatnot. Correct? So, and like all this stuff that can damage the DNA, and our repair system can reverse this chemical DNA. So if there is a DNA damage, let's say here this DNA damage, cells try to repair, they are very, very well studied, extensively studied, at the same time poorly understood. Correct? Area of research is DNA damage and repair. Thousands of people are working. Correct? So one of the ways repair is that it removes the damaged base here. So you create a gap, and now you fill up this gap and ligate. Does it make sense? This is called base excision repair. So you remove the base, hydrolyze out the damaged base, and fill it and seal it. Does it make sense? This is called base excision repair. There are nucleotide excision repair. Just remove the nucleotide or just remove the base. Variety of repair system, including recombination. Recombination also could do to remove a large part of the damaged DNA. Does it make sense? So repair is part of our homeostasis mechanism. Our body is designed to do the repair, but some people are more sensitive to repair than others. Okay. There is a mutation. I give couple of examples. BRCA1. Anybody knows what this gene is?
Yes.
It's a gene which is mutated in familial breast cancer. If some family has the issue of the breast cancer, it's advisable to them that they screen it. If there is a mutation in this particular gene, BRCA1 and BRCA2, these people are in the high risk group. And these particular proteins are involved in repair. That means these proteins are mutated. The repair system is not so good. Does it make sense? So if someone has mutation, analyzed or diagnosed with this mutation, they are advised not to take a lot of the sunbath or correct. Take some precautions so that it reduces the risk. There is a gene called transcription factor called TF2/8. This particular gene is involved in repair. In fact, there is a British population which has a disease called xeroderma pigmentosa. Xeroderma pigmentosa. It's a skin disease. This is certain British population, they have the mutation in their gene, so they are highly prone to this particular skin disease because the repair system is not working. So repair is very, very important. If the repair is compromised, people are at high risk of cancer. Does it make sense? So any gene which are involved in DNA repair is malfunctioning. That means you are compromising the repair. Chances of mutation accumulation will increase and increase to the risk of cancer. These people are sensitive to UV light, gamma radiation, sunbath, mutagens, and all this stuff because they are compromised in repair. So repair is a very important stuff. There is base excision repair. Basically, just remove the base. Sometimes there is nucleotide excision repair, which is called NER. That means the base, remove the nucleotide, correct, and do the job. So just read. Now, what is the molecular basis of mutation? We discussed some of them. Point mutation arise from inappropriate base pairing. So during the DNA replication, a wrong nucleotide is incorporated, correct? And although there is proofreading is also not 100%. So still one can end up with 1 in a million or 1 in a billion wrong nucleotide incorporation. Does it make sense? So that's a point mutation. Point mutation means single nucleotide mutation. We have complete genome or complete gene correct, but there is one critical base is replaced with another base. Correct? So that's point mutation. Mutations would be caused by base analogs. I will give you some examples. Mutations, chemical could induce mutation, insertion of the genome, deletion of the genome, all these could cause mutation. Point mutation sometimes is driven by inappropriate base pairing. Normally, you form A and T, 2 hydrogen bond. That's the best base pairing. Right? They have the hydrogen bonding. At least in the physiological case, this is the best base pairing. But we have studied the base can also go into the ketoenol tautomerism, correct? So if it goes to ketoenol tautomerism, their hydrogen bonding acceptor and donor property changes. So then they are correct. Base pairing ability with other bases also changes. Like TA is the base, but sometimes A can also form weaker, but some pairs with G. So normal DNA you have TA, but if the adenine pairs with G, it will cause a mutation because next round half of the DNA will be AT and half of the DNA will be GC. Does it make sense? So this is a point mutation. Sometimes point mutations are tolerated by the body because it's not so important for the protein, but sometimes it could be problematic. Correct? So this is an example. So TA, AT, half of them will be AG, half of them will be AT, and AG it will be GC and AT. So some population of the DNA will be mutated to completely GC, which are supposed to be AT. These are extremely hard to detect because there is no damage. Correct? These are only detected at the stage of proofreading when nucleotide is incorporating. Does it make sense? But once it is incorporated into the DNA, the repair machinery cannot detect these mutations because these are the same base. How does repair machinery will detect whether it's right or wrong? Does it make sense? So proofreading mistakes causes the mutation. 5-bromo-uracil. Uracil normally pairs with which base? AU, correct? Adenine, correct? Yes.
So when the DNA repair mechanism happens, how does it recognize the wrong? Is it just knock off any bond that's too weak?
If it is a AT to GC transition already done in the DNA, it's hard for DNA repair mechanism to decide. But if there is a damage in the DNA, let's say I will give some examples, some base is damaged, then repair machinery will come and bind here. Okay? Otherwise, repair machinery cannot figure it out. So 5-bromo-uracil is a mutagen. Lot of people use it also in the lab. Basically, this form uracil is supposed to form. Normal uracil is supposed to form a bond with a U type of bond. but it can also pair with G. Correct? So instead of uracil, if someone is used 5-bromouracil, it will convert AT to GC type of transition because uracil can pair with G. Correct? So if the cells are exposed to 5-bromouracil, so you are growing some culture human cell, they are adding 5-bromouracil, and couple of rounds later, we analyze the genome. You see there are lots of changes that happen, basically AT to GC type of transition. Does it make sense? Yes or no? 2-aminopurine. So normal adenine, this is let's draw the way the DNA is drawn. This is number 9 Poisson, and I have to check numbering, but I think this is okay. 6, 7, 8, 9. This is where your base is connected, correct? So what is this molecule? What is this molecule?
Adenine.
It's adenine. Correct? It's adenine. You also call the 6-aminopurine. Which forms base with thymine. Correct? 80 base there. Now, instead of 6-aminopurine, if we have 2-aminopurine, which is kind of adenine, but it's not adenine. 2-aminopurine is a nucleotide base analog, right? It's similar to adenine, but it's not adenine. This 2-aminopurine can form vector thymine, but 2-aminopurine is also capable of forming pairing with cytosine. So if the growing cells are treated with 2-aminopurine, this 2-aminopurine will converted, integrated into the nucleotide triphosphate. Eventually cell will incorporate them. Sometimes it will be AD, which is normal, no mutation, but it can also become a C pair. So if it is mutating, pairing with C, next round, half of the DNA will be GC. So if the cells are treated with 2-aminopurine, it will convert AT to GC type of mutation. Does it make sense? Same is true for bromouracil, AT to GC type of transition. Does it make sense? Yes or no? So adenine, know this bromouracil and 2-aminopurine, what type of mutation they cause. Next one I am going into something. Modification of the DNA. Adenine is the normal base present in DNA, correct? But this adenine sometimes is hydrolyzed or oxidize to corresponding carbonate. So basically a nucleophilic substitution here and then oxidation. NH2 will be replaced with OH and water. Water is reacting with adenine and then oxidation to give this oxo compared to NH2, and this is called hydrogen thing. Now think about this. NH2 is a hydrogen bond donor, correct? Whereas this becomes an hydrogen bond what? Acceptor. So see, just the changes in the group is changing the hydrogen bonding profile dramatically. Does it make sense? So this hypogen thing, so adenine normally put hydrogen bond with thymine. Instead, this hypogen thing, because it's hydrogen bond acceptor now, it forms hydrogen bond with cytosine. So conversion of adenine to xanthine lead to again AT to GC type of mutation. Does it make sense? In fact, in the DNA, if one screens the genome, you will see that many places detect the hypogen thing. Is it clear? So when I was many years ago, I was auditing a class with my postdoctoral supervisor, Linda Rehakans, University of Alberta, Canada. Because I was a chemistry student, I was trying to understand biology. I never took a biology class in my life. So it was a little bit hard, but I was interested to know. She is one of the best geneticists I have ever seen. Okay? By looking at so she takes the bacteria or the virus and treats with some mutagen. Okay? If the cells are treated with mutagen, high dose of mutagen or cancerous drug, most cells will not survive because of the replication problem. Does it make sense? Say, like actinium or an ethidium bromide. You took some cells, treat with lots of ethidium bromide or UV or something which will kill the cells, correct? But what you see that, so let's say you take a plate of cells, whatever the cells, this is full of cells. Okay? Now you treat with a strong mutagen, high concentration. Let's say ethidium bromide. Ethidium bromide or acridine orange. These are all aromatic compounds. Or you can use 2-aminopurine, or you could use bromo-uracil. Any of these mutagens you take. Does it make sense? These are relatively better, but these are nasty chemicals. 99.9% cell will die because they are so bad. Eventually, you will have a plate with 1, 2, or 3 cells or 5 cells surviving. Each cells are special. What is special with these cells? They have some problem. And their problem is a good problem here because they possibly have efficient repair system. Does it make sense? They have extremely efficient repair system, or they are bypassing the mistake. Some of these problems. So these few cells, if you sequence, you will find the mutations in some of the repair genes. Some of the genes are whatever, correct? And those genes, you will think that they are involved in repair. This is genetic thing, correct? Or any type of organism You subject to extreme condition. Correct? Some of them survive. Those survivals are the odd ones. But they survive because they survive in that natural environment and they have certain special things. Does it make sense? That's how you can detect, and she finds that mutations in the proofreading active site and DNA polymerase, variety of stuff. Yes.
So with hypoxanthine or any other analog, what is the complement to it? Like if there's replication going on, it can't repair it because you said it doesn't detect it. usually?
Hypoxanthine is a modified nucleotide, it could be repaired by that.
Okay.
Hypoxanthine has some modification, so repair system does recognize hypoxanthine.
Okay.
But still, because this is so common, and this is done just by water and oxygen, right?
Then for 2-aminopurine, what?
2-aminopurine is external agent, it's not present in our natural environment.
Oh, okay.
Until and unless we add 2-aminopurine, cell do not have 2-aminopurine. But this is natural because we have adenine, we have plenty of water, plenty of oxygen. So water is a major mutagen. Correct? That's what is Linda reactant, the geneticist says that. Water is the most important mutagen we have has. Correct? But if we drink a lot of water, we don't think it's mutagen, and I also don't think it is mutagen, but it could cause the mutation followed by oxidation. Does it make sense? So read some of these mutations. There are lot of alkylating agent, lot of chemical response. Correct? Nitrous acid, active, reactive phosphate, phosphonate, correct? Pesticides. We use a lot of pesticides, weed killer in our garden. Correct? How do they kill? Sometimes they damage the earth. Actually, many of them are basically reactive molecules. As soon as the plants and animals are exposed, insects are exposed, they die. Why they don't kill us? Any answer? Pesticides? If pesticides can kill pests, they are also equally potent to kill humans. It's a matter of both. Alright? Sometimes they little bit tested preferentially. So all these pesticides, chemicals, all these fancy vegetables which are not getting rotten because they sprayed with pesticides or genetically modified, all these pesticides are coming to us. I don't have time to tell the story, right? But it's a big, big problem, especially in third world countries. Remind me next class. I'll tell a story. Okay. Pesticides, fertilizer, and environmental stuff, epigenetic changes. These are all my favorite topics, and I stay away from the study. So next class, read this section, some of the alkylating agents, how it does, and next one is, I think, immunology we are not getting, so we kind of stop here for the mutation.